this technique, glass cover slips are functionalized with a mixture of inert and biotin PEG molecules. A flow cell is created with this functionalized surface and placed on a turf microscope. Biotinylated rolling circle templates are attached to the surface using multivalent streptavidin. When the substrates are replicated by addition of replisome proteins and nucleotides, the resulting double-stranded DNA is extended with laminar flow and imaged in real time using an intercalating dye.
Hi, I'm Nathan Tanner from Antoine Van Oijen's lab in the Department of Biological Chemistry and Molecular Pharmacology at Harvard Medical School.
I'm Joe Loparo, also from the Van Oijen lab.
Today we'll show you a procedure for fluorescence imaging of DNA replication in real time at the single molecule level.
We use this procedure in our laboratory to study coordinated DNA synthesis activities of individual replication complexes. So let's get started.
To begin this procedure, anneal the biotinylated tail oligo to the M13 single-stranded DNA by adding a 10-fold excess of oligo in TBS buffer, heating to 65 degrees Celsius, then turning off the heat block to allow slow cooling to properly anneal the primer. Now that the primed M13 is ready, add it to a mixture of 64 nanomolar T7 DNA polymerase and T7 replication buffer containing DNTPs and magnesium chloride, and incubate the reaction at 37 degrees Celsius for 12 minutes. At the end of the incubation, quench with 100 millimolar EDTA. Next.